All right. Hello and welcome, everyone. We're so glad that you could be here today. Thank you so much for joining. My name is Stephanie, and I will be your co-host for today's webinar. Now, today's webinar is part of our larger BrainHQ Academy series, where we talk about the neuroscience behind different topics. Today's topic is brain chemicals, the chemistry of your brain. So let's go over the agenda for today. All right. So starting off after the agenda for today, we are going to be doing a game via a poll here in Zoom. More on that when we get to it. Following that, Dr. Mouna Attarha is going to give a primary presentation on brain chemicals in the brain. Mouna, you want to hop on and say hi?
Excited to be here, everyone.
It's always great to have you back. For those of you who have not seen a presentation by Dr. Attarha before, she earned her PhD in cognitive science, and she's been a senior scientist at Posit Science since 2016. She leads NIH-funded clinical trials investigating the neurochemical changes that occur by training on BrainHQ. And she also manages several trials looking at how BrainHQ exercises improve cognitive performance in healthy adults and in those with mild cognitive impairment and early dementia. So perfect person to have talk about chemistry of the brain today. Now, joining us for a discussion is going to be Dr. Michael Merzenich. Mike, you want to hop on and say hi?
Sure, it's nice to be with you. Uh-oh, just shut myself off. It's nice to be with you all, and I'm sorry we can't be with you in person. Maybe sometime we can do that.
Oh, that'd be a wild idea, getting everybody in an auditorium and doing it seminar style. That would be fantastic. But for those of you who aren't familiar with Mike, Dr. Merzenich is the co-founder of Posit Science, our chief scientific officer, and the original brain behind BrainHQ. For nearly 5 decades, he has been a leading pioneer in brain plasticity research and has published more than 150 articles in leading peer-reviewed journals. So it's a fantastic combo that we have here talking today about brain chemistry. Following the discussion between Mouna and Mike, we are going to have a live Q&A. So if at any point during the presentation today you have a question, you can click on the Q&A button on the Zoom control panel, and a window will pop up where you can type in your questions. There are a lot more of you in the audience than there are 3 of us here leading the webinar today, so we aren't going to be able to answer every question, but I will be doing my best to curate those questions to get as many of them answered as we can. One other thing is that I wanted to note that today's webinar is being recorded and it will get posted to our YouTube channel in the coming days. If you'd like to be notified when that video goes live, you could head on over to youtube.com/brainhq and subscribe there in order to receive a notification. Also, everyone who is registered for this webinar will also receive an email sometime next week or the following week with follow-up resources if we include any, and also a link to the replay on YouTube. All right, so let's go on to the activity here. So we are calling this the brain chemical match game. And what I'm going to do is I'm going to go ahead and launch a poll here for you all. Now, this is going to be 3 questions, and it's multiple choice. And we want you to go ahead and give your best guess as to what you think the answers are here. So the first question, you're learning a new dance routine and finally get the steps right. Which chemical helps your memory and learning? Is it 1, acetylcholine, 2, serotonin, or 3, dopamine? Second question, you win a board game and feel a rush of excitement. Who's celebrating? Is it acetylcholine, serotonin, or dopamine? And then final question, you hug a loved one and feel warm and calm and safe. Which neurotransmitter is at work? Is it acetylcholine, serotonin, or dopamine? Now, a lot of these chemicals will interplay with one another, but we're sort of trying to ask you to emphasize the chemical that you think is most predominantly associated with the action included in that question there. So it does look like we have quite a few people have already chimed in, which is fantastic. We're going to give it another minute here just for people to get their guesses in. And it's okay if you're not sure, go ahead and give a guess either way. Part of the presentation today is going to be going over these questions and what these different neurochemicals do. So it's okay if you don't know anything right now. All right, Mana and Mike, do you want to hop on so that way we can talk about some of these results here? All right.
Yeah.
So we're seeing
It's fun to see.
yeah. So the first question, you're learning a new dance routine and finally get the steps right. Which chemical helped your memory and learning? Now it
Well,
looks like most people went and guessed acetylcholine. So let's see if that's right.
well,
And it is.
well,
Uh,
actually, dopamine is a pretty close second
Very close second.
because dopamine is telling your brain basically that that's just happened was important and you should save the answer. And so it's also involved pretty closely in learning and memory.
For sure. All right. And I think that also is reflected in the results here where acetylcholine was selected the most and then dopamine got second. So
Right.
not bad, folks. All right. Next question. You win a board game and feel a rush of excitement. Who's celebrating? Is it acetylcholine, serotonin, or dopamine? Well, you all got it right again. It is dopamine.
Oh, it looks like we have to expand the window to see all the questions and the answers. So
Oh,
if
I'm
you're only
sorry.
seeing part of it, just expand that lower right-hand corner, and you'll see all the
Got it.
responses.
Sorry. I have a very large screen, so I've got a lot of real estate
I'm
here.
on a laptop. Yeah, that makes sense.
Yeah. Yeah, you might need to scroll down or resize that window if you're having trouble seeing those results. All right, and Mike, can you talk a little bit about the rush of excitement you get and why that's dopamine that we're feeling the most?
Well, dopamine basically is a chemical that's controlling reward, and it basically is released whenever we get the answer right. So when you play BrainHQ, we're releasing a lot of dopamine, and that's by intention. We're trying to exercise this machinery because keeping this chemical machinery in good shape is one critical aspect of keeping your brain in good shape. So it's a key chemical in controlling learning.
Absolutely. And then final question here, since we already have acetylcholine and dopamine as our first two answers, we all know that the third one's going to be serotonin. And serotonin is a wonderful neurotransmitter. Mike, can you speak to that a little bit?
Well, when you're really low, that is to say, when you're thinking about whether life is worth hanging in there for, basically serotonin is at fault, and that is to say, you're low in serotonin. And when you're a happy person, serotonin is provided to you in your brain in generous supply. So it's really a good idea to keep your serotonin production up in your brain in your life because it's all part of being a happy clam.
Absolutely.
This is a
All right.
smart group, I have to say.
I know everybody got it right, or
Fantastic.
the most people got all those answers right, which is fantastic. All right. Well, I think with that poll out of the way, I'm going to go ahead and toss it over to you, Mana, if you want to take over.
Great. All right, so let's talk about how brain chemicals shape our brain and behavior. So, when we think about how the brain works, we're not talking about what single neurons are doing in isolation. There are instead these very large networks of neurons that have a big impact because they send signals that can shape how the whole brain functions. So, if we take acetylcholine, for example, these neurons help with synaptic plasticity or the brain's ability to strengthen and weaken connections based on our lived experience. They're also, of course, critical for attention, learning, memory, and higher order thinking skills like decision making. Then there's dopamine. You've probably heard of dopamine in the context of reward and pleasure, but they do more than just reinforce behaviors that feel good. They keep us motivated, moving towards our goal, and they also play a very large role in motor control. And then finally, serotonin. Serotonin neurons are heavily involved in mood regulation and emotional stability. They also influence social behavior and help us stay emotionally resilient in the face of stress. And this is intended to be a very, very brief, high level refresher on synaptic neurotransmission or how neurons communicate with each other. And most communication between neurons occurs at a specialized structure called a synapse. And a synapse is an area where two neurons come close enough together that they're able to pass chemical messages from one cell to another. So the neuron where the signal is initiated is called the presynaptic neuron on the left, while the neuron that receives that signal is called the postsynaptic neuron on the right. And in the presynaptic neuron, there are chemical neurotransmitters that are packaged into small sacs called vesicles, and each vesicle contains thousands of neurotransmitter molecules. And so when the presynaptic neuron is excited by an electrical signal called an action potential, it causes these vesicles to fuse with the membrane, release their contents into the synaptic cleft. And once they're in the synaptic cleft, neurotransmitters interact with receptors on now the postsynaptic membrane. They bind to these receptors and can cause an action to occur in that postsynaptic cell as a result. And this action may involve increasing the likelihood that postsynaptic cell will become activated and fire an active potential. It also itself and then also can decrease it, inhibition in that case. And then eventually the neurotransmitter molecules must be cleared from the synaptic cleft. And some of them will simply, drift away in a process called diffusion. In other cases, the neurotransmitter is taken back into the presynaptic neuron in a process called reuptake. And once back inside, that neurotransmitter can be recycled and reused. And then in other cases, enzymes break down the neurotransmitter within the synapse. So that's called enzymatic breakdown. So as we go through that was a lot of nerdy stuff and terminology, but as we go through this presentation, keep in mind that you can alter synaptic neurotransmission by damaging it. You can alter it by commonly used medications that keep those brain chemicals in the synaptic cleft for longer, either by altering the enzymes that break it down or by altering the reuptake process. And then you can also change the plasticity of the system itself so that there are more of those vesicles with neurotransmitters that are available for packaging and release. All right, so this schematic here looks complicated, but just focus on the right side for now. It shows us why neuromodulatory systems have such a big impact. What you see on the right are these several color-coded locations of neuromodulatory nuclei, which are essentially very dense clusters of neurons. So for example, we have the nucleus basalis there in yellow as one of the nuclei for cholinergic neurons. There's the substantia nigra and the ventral tegmental area in red for dopaminergic neurons. There's the raphe nucleus for serotonergic neurons, and so on. There are many other nuclei. And what you see on the left now are the very long axonal projections from those nuclei, but you'll notice that they aren't just going to just one region of their brain, but they're extending across a vast range of cortical regions and layers that are associated with cognition, behavior, mood, and motor function. So neuromodulatory systems make a big impact because they broadly regulate and adjust the activity and plasticity of large neural networks. And in the case of acetylcholine, what we know is that you can manipulate the cholinergic system to change cognitive performance. So if you stimulate the cholinergic system, you can improve your attention, learning, memory. And if you impair the cholinergic system, maybe you lesion it, maybe you block its action with a drug, then you would score worse on measures of attention, learning, and memory. And because these sorts of cholinergic manipulations are often invasive, most of this research has been done in animals. But I'm going to give you some of the basic science done in this area before we move on to work in humans, because it really does nicely set the stage. All right, so you take a group of healthy rats and you give them a memory task, and they're going to score really quite well on that memory task. They'll be accurate about 75% of the time, which you can see in that leftmost black bar. But if you then give those same rats a drug to impair their cholinergic system and deplete acetylcholine, they'll now perform that exact same memory task, the one that they used to perform well on, no better than 50-50 chance. They can't do it well anymore, which is what we see with that very short second black bar. But now, if you give that rat a drug called donezepel, which is what we also give to humans to increase acetylcholine by keeping it in that synaptic cleft for longer, what you'll see is that memory improves almost back up to 65%, 70%. So what we're seeing here is that we can impair memory by decreasing cholinergic signaling, and you can improve memory by increasing cholinergic signaling. So studies of this nature really speak to the validity of this brain chemical in supporting memory and, of course, other cognitive functions. We can do something similar with dopamine. So rats are highly motivated to work for food that they like. So if you train a healthy rat to exert some effort by, let's say you train it to rear up on its hind legs to press a panel with its nose for a very delicious strawberry milkshake, it will absolutely do it. It'll be motivated to get that strawberry milkshake. A rat that's on no meds, so which we can see in those two white bars, will press that panel about 160 times within 30 minutes for their milkshake. But if they don't want to work for their food by rearing up, there's also standard rat food in the room with them. And they have the option to eat those, which they choose to do some of the time, but not too often when they're healthy. They rather work for, the greater reward. But now, if you give a rat different doses of a drug that impairs their dopaminergic system, what you'll find is that their willingness to exert effort for the milkshake steadily declines. So, at 8 milligrams of the drug, that's those solid black bars on the right, the rat is now pressing the panel about 85 times in 30 minutes. That's down from 160. And their intake of the easy food, those pellets that they didn't have to work for at all, has steadily increased with increasing drug. So this result speaks to the general finding that dopamine influences motivation and effort, which is a behavior that we often see impaired in various medical conditions that involve damaged dopaminergic systems. And then similar experiments have been done for serotonin, showing that we can cause animals to have a negative cognitive bias, meaning that the animal will interpret ambiguous information, so something that is fully neutral, as more pessimistic than it really is. And negative biases like these are, of course, common across a number of neuropsychiatric disorders. So, as I implied, impairments in these neuromodulatory systems, while not going to be the single cause of human disease, do have at least some influence via their brain-wide reach. And they've actually historically been the target of pharmaceutical treatments. So, in the case of acetylcholine, we know that the cholinergic system is impaired in various neurodegenerative diseases like dementia, and that patients are commonly prescribed cholinesterase inhibitors like donazepam to keep acetylcholine in the synapse for longer, just like the rats were given. We know that the dopaminergic system is impaired in psychosis like schizophrenia, and that patients may be given dopaminergic meds that block dopamine receptors to reduce excess dopamine signaling, like haloperidol, for example. And we know that the serotonergic system is impaired in mood disorders like major depression, and that patients are often given selective serotonin reuptake inhibitors to keep serotonin in the synapse for longer, like in the case of Prozac. So these and other neuromodulatory systems have informed how we've designed the exercises on BrainHQ. So I'm going to use Hawkeye as an example, and Hawkeye trains your brains to quickly find the item that's different from the others. And so what you're going to see is a bird highlighted here that looks different from the others. And the bird can briefly appear anywhere on the screen, and your job is to report where it was. So go ahead and do this along with me. Where it appears, you select it, and then you get, Stephanie, were you able to hear the sound on that one?
Yeah, there was a very faint beep.
Okay, there's a faint beep. Okay. So let's talk about this task from a neuromodulatory standpoint. When that bird appeared on the screen, that information was sent to your visual system and then it was quickly taken away. And your brain had to do the best it could to quickly and accurately process what it just saw, given that it was only available for less than a second. And in order to do its best, your brain had to pay attention. That causes your brain to pump acetylcholine. It's the brain's pay attention chemical. And acetylcholine tells your brain, be ready for change because something interesting is about to happen that you need to remember. And then once the bird disappeared, you had that spotlight that you can move around the screen to select the location where the bird appeared. So now you're engaging your memory and reporting your answer. And when we got the answer right, we got a little reward, that pleasant ding sound. And getting the answer right for something that was hard and hearing that ding actually causes your brain to release some dopamine. That's the brain chemical involved in motivation and reward. So, dopamine is telling your brain, whatever you just did, that worked. So, reorganize your brain a little bit to do more things like that. And I'm going to go ahead and play a few more trials here. Try to do these along with me. And as I go through this, notice that there are a whole lot of birds on the screen, and all of them, all those distractor birds, are vying for your attention. They're trying to distract you away from the target bird. And across each of these turns, where that bird appears on the screen will be unpredictable. And on each turn, its location is going to surprise you and release another brain chemical we've covered in previous webinars called norepinephrine. And then finally, completing your training doing your best on it, being really engaged with the training exercises, releases brain chemicals that provide a feeling of accomplishment. Okay, so this is going to be a mini recap. What do we know so far? We know that there's extensive evidence in animals showing that brain chemicals influence cognition, mood, and behavior. We've observed these kinds of effects in rats, in pigs, mice, monkeys, and cats. So it seems to be a fundamental property of how brains work. We know that cognitive training in animals can actually change neuromodulatory systems, but this has historically been very difficult to look at in humans until somewhat recently. But the good news is that we now have the technology to non-invasively measure brain chemicals to find out if neuromodulatory systems are plastic in humans, just like we know that they are in animals. So we can measure cholinergic function in vivo using a radioligand called FeBOV with PET imaging. And what FeBOV does is it selectively binds to something called vesicular acetylcholine transporter, which is uniquely expressed by cholinergic neurons. And that transporter is shown here on the right. FeBOV is binding to that salmon-colored transporting unit that's holding a red acetylcholine compound. And we recently completed an NIH-funded study called INHANCE, which uses FeBOV PET to see if cognitive training can improve the cholinergic neuromodulatory system. This was a double-blind randomized controlled trial randomizing older adults either to BrainHQ or to an active control of computer games like Solitaire and Candy Crush. The primary outcome, or how the trial evaluated success, was FeBOV binding within a brain region called the anterior cingulate cortex. And the anterior cingulate is really important for attention, learning, memory, and executive function. And then on an exploratory basis, we included other brain regions that we know degrade with aging and with neurodegeneration. So like the hippocampus, for example. And here are images of the intervention and the active control. I'm sure the images on the left are going to look familiar to you. But the intervention included two training exercises. The first one is called double decision, which was shown previously to reduce the risk of dementia by up to 48% in a previous study. And the second exercise is called freeze frame below that, which was designed to engage neuromodulatory centers in the brain, and which has been shown previously to actually work synergistically with double decision. And then the active control group had spin-offs of games like Solitaire. So this is a game where you move the cards in the 8 foundations to the top by suite. And then a game that's kind of similar to Candy Crush, where you eliminate the bricks by clicking them in groups of the same color. And then regardless of which group you were randomized to, participants had to train on the program for 35 total hours over the course of 10 weeks. So a short training period, 10 weeks, but getting all 35 hours within those 10 weeks. So they did about 30 minutes a day, 7 days a week. And here are the very lovely core results. Those who were in the BrainHQ group, so did speeded brain training, showed a significant increase in FeBOV binding in the anterior cingulate cortex. There was also a notable between groups effect in an important subregion of the anterior cingulate that's associated with executive function. And there were significant increases in other brain regions that support memory. So the hippocampus, which we know declines with age and neurodegeneration, as well as the parahippocampal gyrus. That wasn't true for the active control group, though, playing solitaire and Candy Crush spin-offs. There were actually no differences in feedback binding between when they started their games and when they finished those games 10 weeks later. So this shows us that a specific type of cognitive training, one that trains speed and one that heavily involves, what we know about neuromodulatory function, can increase cholinergic neurotransmission in areas of the brain that support attention, memory, and executive function. And this trial represents really the very first demonstration of cholinergic system network plasticity now in humans for the first time. And here are the PET images showing that changes in Febov binding within the anterior cingulate. And what you can see here is that those warmer colors, the reds and the oranges, reflect greater increase in acetylcholine signaling after training compared to baseline. So there's a lot more red and orange in that intervention group on the left than there is in the active control group on the right, according to their PET scans. And these are nearly all the brain regions we looked at. The taller the bar is, the more Febov binding there was in the brain for that region. Black is BrainHQ, gray is the active control. And the increase in Febov ranged anywhere between, 0.5% increase, like in the parietal lobe, all the way to a very large 5% increase, like in the hippocampus and the parahippocampal gyrus. So to review here, brain chemicals play a key role in cognition, mood, and behavior. We designed BrainHQ exercises with these systems in mind. We ran a gold standard NIH-funded clinical trial that provides the very first evidence in humans that neuromodulatory systems can plastically adapt and improve within 10 weeks of speed training. And what's great about this is that we now have a biological mechanism of action for cognitive training. We're no longer talking about a cognitive construct or process. We're talking about an actual biological change that aligns with the standard pharmaceutical framework for drug development. And then lastly, we know that that drugs like cholinesterase inhibitors like donazepam, Aricept are in widespread use in patients with mild cognitive impairment or with, Alzheimer's disease, but they've only shown modest short-term benefits to cognition. And it differs from the speed training here, which is instead restoring the amount of acetylcholine that's released during normal signaling, as people naturally need it when they interact with the world. And that would work differently in the brain because it's endogenous, right? It's natural. It may be, and that may be driving greater clinical benefit across patients who have impaired neuromodulatory function. So all of this leads us to our second round of the brain chemical match game. Stephanie?
It sure does. So first, Mouna, fantastic presentation. Uh, Absolutely wonderful. Let's move on to the surprise round two of the chemical match game. Let me go ahead and launch that poll this time. All right, poll has been launched. You all should be seeing a little window pop up on your screen. Again, there are 3 questions, and you may need to scroll down to see them all. But same as last time, we're going to ask 3 questions, and you have to assign which of those neurotransmitters best answers that question. So first one, you're out in nature, breathing deeply and feeling peaceful. Which neurotransmitter is helping you balance your mood? After that, we've got you're trying to remember where you left your keys. Which chemical is helping your brain to recall that information? And again, this is we're asking for the neurotransmitter or neurochemical that we're sort of asking for the most emphasis on in order to answer these questions. A lot of these chemicals do have a lot of crossover, but which one is predominantly the one that would best answer that question? And then finally, you earn a significantly higher score on round 2 of this brain chemical match game, which neurotransmitter gives you a sense of accomplishment, which great question, but also so many of you got so many of those questions right during round 1. All right, so you're out in nature, breathing deeply and feeling peaceful. Which neurotransmitter is helping you to balance your mood? As you all had most of you had all correctly guessed serotonin there. All right, and then the second question, you're trying to remember where you left your keys, which chemical is helping your brain recall. Most of you, once again, got this right with acetylcholine. And then the final question, you earn a significantly higher score on round 2 of this brain chemical match game, which neurotransmitter gives you the sense of accomplishment. Although most of you probably got a lot of those questions right on the first round, you all understood the assignment here and mostly guessed dopamine as well. So yeah, fantastic job, everybody in the audience.
Yeah.
Congratulations.
They did great.
Yeah. All right. So I think I'm going to go ahead and stop sharing my screen here and stop sharing the poll. Mike, if you want to hop on and have a little chat with Mouna about her presentation and all those wonderful findings.
Mike, I have questions for you. So, how common is it for these neuromodulatory systems to decline with age?
Well, they all do if they're not taken care of. They need exercise. And actually, we conducted animal experiments in which we basically manipulated the serotonin system. Serotonin, very little serotonin was expressed. The neurons that produce serotonin were very inactive, and there was very little serotonin introduced out into the brain. This would be what you'd see in someone that's really depressed, really suicidal, really down. And we train the animal, and we train the animal with training that's akin to what we use in BrainHQ. We weren't able to do the human experiment, but we were able to do this experiment in an animal. It turned out it was a rat. Completely recovered serotonin.
Were those the auditory exercises?
The training was auditory. They were listening-based exercises. That's right. And other scientists did the same thing with dopamine. They basically set up a condition in which dopamine was in decline. There was little dopamine being produced. It was actually a model of the decline in dopamine that leads to Parkinson's disease. And what they saw was that they could pretty completely recover dopamine levels and production and availability in the brain by training the animal. In this case, it was a monkey. And almost certainly we can accomplish that in humans. We don't know for sure, but we're certainly the training that we designed or that we apply in BrainHQ was designed to duplicate what happened in those monkeys.
Mm-hmm.
And then a similar experiment was done in a fourth chemical that's really important also, and that's norepinephrine, which you talked about. Is also involved in your attention. It basically is turning on the lights in the brain. It's increasing the excitability of the brain all across the brain. And again, scientists did studies, a friend of mine, in which he demonstrated how you could train the animal, again, this was a monkey, and turn up the lights. And so we know, but the missing ingredient in all this was acetylcholine. Acetylcholine is really a key player, right? And there was no clear animal experiment, and there was absolutely no human experiment. We just couldn't see how we could get to measuring it. And now you've done it. That's been done now in this beautiful control trial. And that's so important. I just want to say, I'm talking too much, but I want to say one more
No,
thing.
this is perfect.
When I was a scientist that first began to focus on these things, that is to say, the way brain deteriorates in aging and what you might be able to do about it, the predominant religion that scientists held was that the decline in acetylcholine was probably the primary basis for decline to dementia. And then what happened about 30 years ago is that new discoveries were made about amyloid bodies, about these little poison pills that are created by the brain in Alzheimer's disease. And scientists and drug companies began looking at them as the answer. The creation of this pathology is the answer to the progression to dementia. Now, even when they discovered that there were people that had Alzheimer's disease, all of the symptoms also, but had almost no amyloid bodies. Or there are people that had lots of amyloid bodies and didn't seem to have anything really terribly wrong with them. They still fundamentally held to the amyloid body hypothesis, you could say. They thought that these plaques and tangles in the brain were accounting for it. they're not accounting for it.
So you can have a lot of pathology, but no overt behavioral
Exactly.
systems
That-
of memory loss. And you can
yeah.
also have virtually no pathology and significant
But
memory loss behaviorally.
what I think the scientist actually says and has said for a long time is that pathology expressed in the brain is a consequence, an effect of a decline in these chemicals and predominantly of acetylcholine.
Yeah, and I want to give people a sense of like what the natural decline is if you don't exercise your brain. So just
Right,
using acetylcholine, for example, we know in the literature that there's a 2.5% decline in cholinergic signaling from every decade of life, actually starting between when you're in your 20s.
right.
And
Yeah.
then since acetylcholine is crucial for cognition, this decline can contribute to some of the everyday forgetfulness we associated with aging, but the
Oh,
loss isn't inevitable, right? In the ENHANCE trial that we found there was a 2.3% gain that offsets the 2.5% loss per decade. So that's a meaningful change. The system is becoming 10 years younger after training for 10 weeks.
yeah, and that's just the beginning. I mean, you think about how you would adjust your everyday life and how you work across the period of life in training your brain and exercising your brain. You go to the gym every so often, right?
Yeah.
And you basically take care of it. You make sure that you're in good shape,
Mhm.
right? Then the decline doesn't have to happen. That's what the implication of this. The decline does not have to happen. And all of that other emergent pathology does not have to occur, right? So there's another really interesting aspect of this in this specific study. You looked in the anterior cingulate cortex. Now, this is nerdy stuff for the audience, I'm sure, but
But
this,
they're on it. They're great.
yeah, well, this is also an area that's really controlling the way the brain is signaling adjustments in your body. So there are adjustments in what is called the autonomic nervous system and also adjustments in hormonal release, which moment to moment as you go through your day is making the adjustments of how your organs are adjusted to do whatever your brain is demanding your body to do. If you keep this machinery in good shape, you'll live longer.
Mhm.
You'll live longer. So it's not just keeping your brain in shape. You're also contributing to keeping your body in shape.
Yeah, brain controls how the body
The
functions.
brain is controlling how your body is functioning. And you want to keep that body really, really good at making these adjustments to what the brain is asking it to do, right? I mean, that's one of the main things the brain has to be up to. And that's the area that you reported in this study, that you recorded this really nice change.
Yeah. So Mike, in terms of brain chemicals, is more always better?
Uh, No, there can be you can overdo it, and
Yeah.
you can and also you can exaggerate the way the chemicals are released in specific behaviors. For example, you can exaggerate the way dopamine is released that rewards you, and that can be destructive because it might be rewarding you for the drug or alcohol that you're addicted to, or for
Mhm.
the behavior that you're addicted to, right? So there's a dark side as well as a bright side. For the most part, in the case of acetylcholine, it's positive. And for the most part, but you can dysregulate it in either direction, mostly negative.
Mostly negative. Yeah. So what I'm hearing is that the brain depends on fine-tuned chemical control to stay
It
on top.
does depend upon it. And it's actually designed to keep itself in good shape. If you just use your brain effectively and engage your brain in effective exercise forms and engage your brain effectively in life, it's designed to keep itself operational.
Yeah.
And one
But
of the ways it keeps itself operational is it keeps making these critical chemicals, keep producing them on a level that is required for your successful operations.
yeah. It's interesting you mentioned dopamine, though. I mean, too little dopamine is associated with Parkinson's. Too much dopamine in
Right,
certain regions has been associated with psychosis. So, yeah,
right,
balance is really key, especially for dopamine.
right, right.
Why are drugs that target these systems, so Aricept, Prozac, various antipsychotics, why do you think they are only partially effective?
Well, for one thing, they do nothing to contribute to the production of the chemical. And the brain basically, and as you age, is producing less and less acetylcholine, period. They have nothing to do with the distribution of the chemical. So when you engage the machinery that's producing acetylcholine, this critical agent, basically you're also improving the elaboration of the way the machinery is delivering this chemical to the brain. It diselaborates in aging. It's just not doing as good a job of getting the chemical to the places that it's useful. And now Aricept, what it does, or the drugs that have applied that you might be prescribed or that people are prescribed, what it actually does is it blocks the uptake. It blocks that destruction of the chemical that's coming out of the synaptic cleft, right? So you release the acetylcholine and it's blocking its destruction.
Yeah.
And basically you have more that's circulating around, right? But you're not dealing with the fundamental problem. The fundamental problem, the brain is just not making very much of it. And if you don't do something, it'll make less and less of it. And also, there's another thing it does. When it sees the level is higher, it actually turns down the amount it makes. So there's nothing from the point of view of your brain health, it's not necessarily a very useful thing to take these drugs, even though they help you temporarily, 6 months, a year, you have a little better memory. But they're not really fixing anything.
Yeah, so cholinesterase inhibitors keep acetylcholine in the synaptic
Right.
cleft longer, right, through changing enzymatic breakdown process. But your brain eventually adapts to that very steady availability. And so
Right.
the release of acetylcholine isn't being paired with meaningful events in your daily life or meaningful things to remember. So I think that's why the ENHANCE study is pretty exciting, right? We have that packaging and release of brain chemicals linked now with real world events as they're supposed to be. It's endogenous and there's more of it.
Another exciting thing that you saw in the ENHANCE study is that if you just look at your performance on a specific task, and the specific task was double decision, that's the task that we know is associated with, or we see in another control trial, a reduction of the probability of progression to dementia over the 10 years after training occurred. I mean, that's amazing. 10 years later, fewer people are demented. That's amazing. By the way, you can't find trials done in a controlled way like this that show that with any drug. Now, in relation to that, it's probable that we can keep drugs alive a lot longer and a lot more effectively when we train more completely and train more completely specifically to increase the levels of these critical chemicals. And that's what we're trying to do with BrainHQ, and that's what we're working continuously to try to improve what we do. And one thing you saw is that if you just look at the performance on the single task, double decision, it actually, your final performance actually it correlates with the state of acetylcholine availability in your brain. So you can actually
we can take your double decision score and predict how much
acetylcholine you have, which is a terrific thing. It means it might be a relatively simple, almost free way to determine how things are going on inside chemically in your brain, which is absolutely unprecedented. So it's a great discovery. It's a wonderful thing.
And Mike, do you think the cholinergic system is also plastic in people who are maybe at risk for dementia? Maybe they have mild cognitive impairment? Or is it only going to be plastic in a healthy adult?
It's going to be more plastic.
It's going to be more plastic in the person with MCI.
Right. I think it's going to be more plastic. I think you're going to see greater effects and greater impacts when proportionally.
Why do people with MCI or, who are at risk for dementia have greater plasticity?
Well, it has something to do with the starting point that you're at. I think you have, and I think that a relatively small difference can have a relatively big impact. But I think you're going to see that the brain can still produce more acetylcholine at any age, as long as it can be behaviorally engaged. The critical thing is that a person has to be able to get on task and basically in an appropriate way, get on the task in a regular way, in a way that can upregulate the production. And I think that we're going to see, I know that you are trying to study this directly. You're trying to look at this population in a new study that the government has funded. But And I think what you're going to see in those studies is that you have a strong effect in this population, and you're rescuing many people from within it. Because that's what I think you're going to see. I think you're going to bring people out of that condition, and then they keep training. I think you can rejuvenate them so that they're in a safer position from that point forward. Now, one of the things that we've seen when we train people in MCI is we have strong effects in most studies. Not all of them, but in most studies, there are strong effects. But we see that they have to continue to train for those effects to be enduring. And I think that when we turn the training to focus on acetylcholine upregulation, increasing acetylcholine production, we're going to see more enduring effects. But this is all hypothetical, you know. We
Well, we
scientists can guess
will.
a lot. We have to prove it. We have to prove
Well, as you mentioned, I mean, we were just funded by NIH to run another randomized controlled trial. ENHANCE was in healthy older adults. This new trial is going to be in patients who've been clinically diagnosed with mild cognitive impairment. So we'll have the answer in 2 to 3 years.
exactly. Well,
Yeah.
speed it up, Mouna.
I'll get on that. Mike, you've told me many impactful things over the years, but one of the most impactful lessons was that conditions such as Alzheimer's is a natural, expected end state of a brain that's in chronic disuse. So the
Right.
analogy of that is sort of like how low aerobic capacity, like VO2 max or a slowed running speed, is a natural, we know that that's a natural consequence of not physically training your body over the course of decades.
Yeah.
Can you share your point of view with respect to neuromodulatory systems?
It's not a disease to have them go to hell. I mean, I was pardon my French, but they will decline with the disuse, just like your body physically declines with disuse. Your body is demanding it be exercised, and all of the machinery that's creating these chemicals, these critical chemicals that are sort of survival chemicals for you. They need that machinery needs to be exercised. It needs to basically sustain its ability to continue to produce. If anything, you want them to increase their production so that you're in a safe position, always in a safe position, as if you're much younger. And for any older person, any person over the age of 40 or 50, you should be working on keeping them in fine fettle, you know. And we know we have a lot of evidence increasing it. This is a wonderful turn of events that we now know that this machinery can be changed in a very significant way in a relatively limited period of time by appropriate training.
Mhm. And
And
what do you do in your daily life? Just tell me some lifestyle things you do to kind of keep your
yeah,
modulatory systems
yeah,
healthy and in balance.
just live life to the advantage of your brain and how you're behaving and how you're continuously learning and how you're continuously engaging, how you're continuously challenging yourself. And when you need to, or even when you don't need to, just to check up how you're doing, go to the brain gym and exercise your brain and calibrate yourself and make sure you're still in fine shape.
And
And
outside of the brain gym, what are you doing at home? Let's say when you're away from your computer.
it's sort of embarrassing. I my wife calls me a man of 1,000 hobbies, but I believe that the essence of a life from the point of view of your brain is continuously challenging it. You know, you want to live a life in which you're subject to, in which things are always important to you to get better at. And that's what I'm trying to do. Right now, I'm so busy, Mana, making my Christmas presents because
Yeah.
years ago I decided I was going to make all my Christmas presents as opposed to go to the store and buy them. And that
So,
would be an example of the kind of dumb thing kind of a nerdy thing I do to try to keep my brain healthy.
so Christmas presents for all three of your daughters and then all of the
There's a
grandchildren
host
as
of other
well.
people, grandchildren and sister-in-laws and and son-in-laws. Oh, we have a whole herd of them that have to be dealt with.
It's going to be a fun
They're
Christmas.
almost done, by the way.
So we know that, I mean, brain chemicals are very important, of course, but the brain is highly complex. What are some alternatives to the brain chemical hypothesis that you'd
Right.
like to call out?
Sure. Well, a lot of people, there are a lot of notions about uh, what actually accounts for uh, the deterioration in functional brains and the physical deterioration in brains. I mean, there are almost as many hypotheses or notions about it as there are scientists, I think, sometimes. But most of them relate to looking at elemental process. I mean, this is a very fundamental process. The availability of the chemicals that are actually regulating plasticity. And if you cannot regulate plasticity, if you cannot really enable and sustain plasticity, the brain has no way to continuously heal itself and restore itself. And, it has to be continuously improvable. It has to be. You have to sustain this fundamental ability. So I think we have the winner of all of these conflicting notions about what goes wrong in the brain. I think overwhelmingly predominant notion is still the fact that the brain creates these pathologies, not just in Alzheimer's disease, but in other forms of dementia. But all of these pathologies can be linked to how the brain is engaged behaviorally and to its functional operations and to its chemistry. And so I think what we're primarily looking at in these so-called pathologies, where people think about Alzheimer's as a disease, are the effects of the brain basically sliding off the cliff because of disuse or maluse. And what we need to do is we need to protect ourselves from disuse or maluse. That's the primary thing we need to do. And we need to exercise the damn brain. The brain is begging us to exercise. And exercising it in ways that really advantage its health. And that's what BrainHQ is all about.
So with my eye on the clock, I'm seeing a lot of great questions in the Q&A. Stephanie, should we go ahead and...
Yeah, absolutely. So many great questions. So speaking of BrainHQ, sort of picking up where you had just left off, can you talk about, we have a couple of questions saying, hey, I know when we get the ding sound or the chime sound in an exercise, that is there to help our brains pump dopamine to let us know, hey, we got this question right. Congratulations. We want to keep doing that. Can you talk a little bit about when we hear the bonk sound or when we get a question wrong in BrainHQ, is there a negative aspect to that? Does that reduce serotonin or dopamine in the brain when that happens? Can you talk about sort of that design choice in BrainHQ?
Well, one of the ways what BrainHQ is trying to do is it's trying to make sure that you get most of the answers right. We know that if you get most of the answers right, you don't get horribly frustrated. I mean, all of us wish we could do better, but in generally, when you get most of the answers right, you're actually driving positive change in the machinery of the brain. And if you've got all the answers wrong, then it's a real problem. You could go backward, but you don't want to. You want to be in a mode. So what BrainHQ does is it always adjusts difficulty. And it tries to adjust it backwards so you can get to a point where you can actually begin to operate in the task and then creep it forward, move it forward. Of course, that movement it's not going to be immediate. It takes effort. It's a little bit like saying I'm in the gym and I lift a little weight and I find I can only lift up 5 pounds with my right arm. And how long will it take me to get to 6? How long will it take me to get to 20? It's not going to be a day. Right? So it takes an effort to change it because the processes that are changing are relatively slow, and they're relatively and the progression change that really changes the way your brain is operating as a machine. Wow, you might have to rebuild a whole factory.
Yeah, and I want to add to that these exercises keep you at like sort of a sweet spot of challenge, about 80%. If something is too easy, the brain doesn't change. If something is impossible, the brain doesn't change. It really changes the most when it's under a moderate load, a moderate challenge.
It's an ideal place, and that's where BrainHQ is trying to land you, keep you working.
Yeah, it's kind of like a Goldilocks zone almost,
right?
Where you're getting enough challenge for it to be stimulating, but not so much challenge that it's too frustrating and you would just want to stop training.
Now, what happens in your brain chemically when you do this is I'm on a task like this, and I see a certain challenge. It might be a listening task, it might be a visual task. The brain is basically turning on the lights around the task. And basically it's opening up it's making everything that relates to that task more amenable, more ready for change, right? It's doing that by just increasing its activity levels of every alternative answer. And basically it's enabling you to change from your routine way to something better, to something different. And it's rewarding you whenever you make a progression to something better. And so that's what you need to do in everything you learn that you're going to get better at, that you have to go through that process.
Absolutely. All
Fantastic.
right.
What other
So moving on
questions?
to a couple other questions here, because again, got my eye on the clock here. We're already looking like we're going to run over a little bit, if that's okay with you both. Okay, so a lot of people are asking, how can you tell what your own neurochemical levels are at? Like, how can you tell if maybe you're low on serotonin or low on dopamine? Is there any sort of way to do that at home, or do you have to go to a doctor? And what kind of testing would even happen for those kinds of things, if at all possible?
You want to take a try, Mouna? You want me to?
Well, I mean, I imagine that you would feel some of the symptoms that, low mood for serotonin and maybe trouble with memory for acetylcholine and maybe negative attribution bias or negative cognitive bias for dopamine, just all as examples. But with acetylcholine, as Mike was alluding to before, I mean, you could try playing double decision and see what your score is. And then on our website, you can enter in that score and then you can receive, where you stand relative to other people's in terms of cholinergic function. And that, the algorithm that we use for that conversion was, from the ENHANCE trial through the PET imaging scans, linking the PET imaging data with the behavioral data.
And
So
we know that we know, Stephanie, that there's an association between behavioral performance and the expression of these chemicals. And one thing we're trying to do now is we're trying to advance our ability to make predictions about the broader chemical status of the brain on the basis by assessments that we're working to try to create that will tell us the level of serotonin or tell us about the level of norepinephrine or tell us about the level of dopamine. It's probably within our power to do that. And so we're trying to do, we know that they relate. So, but it takes a scientific confirmation and it takes much more elaborate experiments in human brains to be sure. And we need to quantify everything to be sure that we've defined the relationship in a way but I imagine a day not too far in the future when you go to the doctor and the doctor can do simple little tests and know what the chemical status of your brain is in detail.
but
I
it would be a behavioral test. It wouldn't be a neuroimaging experiment like it has to
and
be
it'd be
now.
almost free,
Yeah.
compared to what they now do, which is a multi-thousand-dollar, analysis.
Yep, that's right.
All right. Next question. Going back to BrainHQ, we had a lot of people asking, hey, is there some way that we can tell what exercises in BrainHQ triggers which kind of neurochemical? I think that's an interesting question, but not super straightforward answer. Would either of you like to address that?
All of them demand that you control your attention.
Yeah.
All of them have an element of engagement of remembering. All of them demand that you be on the ball and alert if you're going to succeed on them. And all of those things are expressing these chemicals.
Yeah. All
All
of
of
them
them
give
are rewarding.
rewards.
I mean, when you get the answer right every time you go through a cycle. So that's that every time you do, and that's 80% of the time roughly, the brain is releasing dopamine. So, we're trying to heavily exercise the chemical machinery of your brain. We're trying to do that. And, maybe there are better ways that we could do it, but no one has a better way to this point.
Yeah, so you basically can't go wrong because all the exercises are built with the same principles.
Right. They're built to reinvigorate this chemical producing machinery.
All right, fantastic. And then one final question before we start wrapping up. Thank you guys for your patience and for hanging on here. But a lot of people are interested, especially after hearing about the results of the ENHANCE trial. Are you guys looking for study participants, and how do I get involved?
If you live in Canada near Montreal and you've been diagnosed with mild cognitive impairment or you would like an assessment for it, yes, reach out to us and we will, we can do a full screening with you at McGill University.
We've also thought about the possibility of asking people basically to self-assess and this is, I'm we haven't organized this yet, but we've been talking about the possibility of having people basically assess their performance, measure their performance on double decision, and then keep track of their training across a period of time. If we looked at 1,000 people in training and see if we can relate how much they train and how they train, with what on their personal schedule, with what changes we see in their performance and double decision, since it does seem to index acetylcholine production in brains. Get more and more of an understanding about what people really need to do to not get just 10 years of recovery. 10 years is a big, improvement, but maybe
Huge.
30 or 40 years of recovery of acetylcholine production. That would be a goal.
Yeah, Stephanie, maybe when we send out the webinar recap, we can send the link for where they can get their cholinergic results.
Right.
Yeah, that sounds fantastic.
Right. That would be good.
All right. So I think that is gonna do it for the Q&A portion. I'm sorry, folks. I know we have so many questions left unanswered, but we're trying our best here. Let me go ahead and share my screen and do a little outro for you all. First, thank you all so much for participating. I do want to let you all know that we do have a calendar of webinar or a webinar calendar for our 2026 season. If you want the full calendar, go ahead and send me an email, support@brainhq.com. My name is Stephanie. If you ask for the webinar calendar, I'm happy to send it to you, but here's a little preview. Our next webinar is going to be January 8th. That's a Thursday at 11 a.m. Pacific time, and it's going to be about how to set your training and schedule goals. So for those of you that maybe want to set yourself up with some goals for the new year as part of a New Year's resolution, this webinar is going to be exactly what you're looking for. Other than that, Mouna, do you have any last words that you want to share with our audience today?
It was really wonderful to be on the call. Thanks for the thoughtful discussion. I mean, I hope what we've learned is that, brain chemicals play a big role in our lives. And while they tend to decline with age, that decline isn't inevitable. And these chemical systems maintain their flexibility to really adapt and change and improve depending on how we engage them in our daily lives.
Yeah. Mike, any final parting words you'd like to share?
Well, first of all, I'd like to say great job in explaining this in this complicated subject, Mouna, and
Had a great
always
audience.
fun to be with this audience and the audience of people that really want to learn and understand. That's cool. And take care of your brain. It's the only brain you have.
I love it. All right. Well, thank you all so much. Thank you, Mouna, for your presentation. Mike, for joining us for the discussion. It's always a pleasure having you both on. And thank you once again to our wonderful audience members for your participation and all of your amazing questions. Again, I'm sorry that we didn't get an opportunity to answer all of them. But if you have any follow-up questions, you can always try emailing us support@brainhq.com. All right, I think with that, I will wish you all a wonderful rest of your day, a happy holidays, happy new year, and we'll see you in the new year. Happy brain training, everyone.